my I'll just acknowledge the recording starting then. It was my interest in the biological aspects of breeding that really made me think, well, hang on a minute, if we're going to breed these really rare horses, what do other people that are breeding really rare animals do? And at the time, I'm talking back in the 1990s, I turned to find out what was going on in the world of captive wildlife management with zoo animals, with white rhinos and giant pandas and Siberian tigers and things like that. And you know, there's an awful lot of science that's well published. So that's very much what was the impetus for me to research the topic. I was awarded the Marsh Christian Trust Award Prize by the Rare Breed Survival Trust back in 1998, I think it was now, quite a good time ago. And I was also invited by Lincoln University to come on board and do what originally was an MPhil that turned into a PhD based on conservation biology of Cleveland Bay horses. And for the last 5 or 6 years, I've been involved with the Rare Breed Survival Trust, spreading the word and helping other breeds, you know, particularly rare equine breeds, but not exclusively equines, looking at how they manage their captive populations. So that's my background. What I want to do tonight is to share an overview of the problems that any closed stud book is going to have. As soon as you take a population and you put a ring fence around it and say, right, that's it, we're going to breed within that closed fence, then it will have some inherent problems, and they're common to all of us that manage livestock in that way. So I want to talk about the general problems, but specifically about how we can actually put strategies in place to deal with those difficulties, and furthermore, how we can then carry on and monitor our breeds and secure them going forwards. And to look at those problems, what we need to do is to have a little bit of basic biological understanding. And I want to talk, and this word you'll hear quite a lot, certainly in the early part of this evening, is inbreeding, to then have a look at mean kinship and find out what the difference between inbreeding and mean kinship is, and to have a look at something called effective population size and see how that differs from actual hooves on the ground population size. Of course, the common thing, whether it's Cleveland Bay horses, Dales ponies, Suffolk horses, Eriskay ponies, is they are under the umbrella of the Rare Breed Survival Trust, all on the watch list, which I know has changed in recent years. But the common thing with most of the breeds that I'm involved with, they are now seen as priority breeds by the Rare Breed Survival Trust. And in turn, the Rare Breed Survival Trust reports to government, reports to DEFRA and to the Farm Animal Genetic Resources Committee, who are tasked with monitoring the status of our livestock biodiversity, because government has an obligation to report that to the Food and Agricultural Organization of the United Nations. So the stud books that we maintain on grassroots or by whatever other means we do it, we feed that information annually back to DEFRA, and they in turn analyze the data and report the state of the health of our livestock breeds back to the UN as part of our work towards achieving what are known as the ACI targets, our targets for conservation of biodiversity. Of all the equine breeds on the watch list, all of the breeds, as I say, that I'm involved with, Cleveland Bay horse, albeit it's in alphabetical order, so it sits right below the Cleveland Bay horse. That doesn't rank them in order of importance or genetic problems, but yeah, I'm involved actively with the Eriskay Pony Society and certainly with the Suffolk Horse Society, both of those breeds with active breed management plans. Not only have I been talking to the Dales Pony Society, but I know there are friends from Exmoor Pony Society on board with us tonight as well, and I have been talking with the Exmoor Pony Society. Those of you that are familiar with the watch list will have actually realized that there's no longer a critical list. or critical category of endangerment. What the breeds are now ranked as 3 traffic lights of priority, at risk, and other breeds under the equine umbrella. And part of the reason for that change in the way that things are laid out is to bring it onto a more scientifically acceptable basis, because those priority breeds are actually designated as priority breeds because of their effective population size or their compromised effective population size. And I'll talk about that in a little bit more detail later on. So priority breed and effective population size go hand in hand. I think there are some people that have just joined that perhaps aren't muted. If I can just politely ask that anyone that isn't muted could just mute themselves at the moment. That stops. Thanks. That's just cured that straight away. Thanks very much. So small populations, whether it's Eriskay ponies, whether it's giant pandas, white rhinos, or whatever, a small population will have biological problems, certainly in terms of genetics, because they lose genetic diversity at a much greater rate than a large population. That's fact. There's nothing we can do about the fact that they lose diversity, but what we can do is put strategies in place to try and turn the tap down. We can probably never get it beyond dripping, but we can stop stuff going down the plug hole. And I'm going to show you in a moment just what that process is. And that's the process called genetic drift. And that actually means that although we might maintain the same number of genes in a population, the actual variety of genes that we maintain diminishes over time. And that in turn is very closely linked to inbreeding, and inbreeding itself is very closely linked to a process called inbreeding depression, which I'll talk about in a bit more detail. And that in itself is associated with the appearance of what we might call harmful genetic traits, these deleterious traits, and the loss of genetic fitness. So genetic diversity, what the heck do we actually mean by genetic diversity? Well, if we could strip out all of our DNA and shake it out on the table, I like to think of all of our genes as a tube of Smarties. And if you tip them out on the table, you would find that you've got a big pile of different colored sweets, and that there will be more of some than there are of others. In this case, I've tipped them out in a pile. We've got a lot more red ones than we have the purple and the yellow ones. And right out on the sides of that pile, because they've been color sorted, there aren't very many blue ones and there aren't very many green ones. And if we were to breed from that population of Smarties, the ones that are at greatest risk of being lost and not passed on into future generations are the ones that we got least of, the blue ones and the green ones. The ones that are best represented, the red ones, are at least risk of being lost. And the reason for that is genetic drift. And what I'm going to do is just talk through what might happen to a single pair of genes. Now, single pair might sound contradictory, but we're what we call a diploid animal. If we uncoil our DNA, we effectively carry 2 genes or two alleles at any one position on the DNA. So we've got the dam there represented on the pink side of the chart and the sire represented on the blue side of the chart. And at any one particular point on the DNA, let's just imagine that our sire and our dam are completely unrelated. Okay, so up here at the top of this chart, our dam is carrying 2 copies at this gene, then carrying A and B for this particular gene. The sire is carrying 2 completely different copies for this gene. He's carrying C and D. So altogether, we've got A, B, C, and D all represented and potentially therefore passing on into future generations. So let's mate these 2 animals, do a cross, and let's see what happens. So we were quite lucky in that first cross. We actually got a filly foal out of them. Look, bingo. What's the filly foal made up of? Well, A has passed on from the dam, and the filly foal's got a D, which has come from the sire. So A and D were passed on. Let's do the cross again. Crossing the dam and the sire with A, B, C, and D. This time in this colt foal, the colt foal has inherited B. That's obviously come from the dam. And it's also inherited D, like the filly foal did. So now we've got the same number of what we call alleles. We've got 4 alleles in this next generation, like we had up here, but we've no longer got A, B, C, and D. We've got A, 1 copy of D. We've got B. C isn't here at all. We've got 2 copies of D instead. So in 1 generation of crossing, we lost C. And that's the basic principle of genetic drift, in that we just lose, we can lose markers. To step back and look at the effect of that, let's suppose we started off with 10 different types of genes in that dish, and we do some random crossing over several generations. By the time we get halfway down the chart, we've still got 10 in the dish, but we've only got 6 different colors. And if we carry on crossing on random generations, we've still got 10 in the dish, but this time now there's 9 red copies and there's 1 blue copy, and we've lost all the other variety, all the other variation has gone. So over a number of generations through genetic drift, we might still have the same number of genes, but the variety is completely compromised. Let's put this into a real life context. I don't know if any of you have been involved with parentage testing that goes on when you register your new foals, but the hair sample or the blood sample gets sent away. Used to go to the Animal Health Trust. Now I think most breed societies are sending them off to Weatherby's down in Southern Ireland. But this is the sort of report you would get back. And what they're testing for is what they call against a panel of microsatellite markers. So it's not the whole DNA profile of a horse. That's about 2.4 billion genes. This is against a panel of 17 markers, VHL20, HTG4, and they've all got their own individual names, these markers. And we've got a database that's already got information about the sire and what they were carrying at these various markers, and about the dam. And we then test the hair sample from the foal to see if that foal could actually have been got by those 2 parent animals. A little bit cack-handed, that particular chart, to understand. So what I've done is I've transposed it into this table. So we've got the sire on the left-hand side of the table. These are the markers listed in tabular form. We've got the dam over on this side. Let's work our way down. This is a bog standard parentage proved parentage test. So at marker number 1, VHL20, the sire was carrying these 2 copies, I and M. This is a bit like our chart before where we had A, B, C, and D. We're carrying I and M. The dam is carrying I and P. And when they were mated, the progeny has turned out to be tested as M and P. Now, is that possible? Well, yeah. M came from the sire. P came from the dam. The progeny is M and P. If we go down to the next marker, quite interesting that the progeny, when it's tested, has took 2 copies of the same allele. It's what they call homozygous. Homozygous meaning both the copies are the same. Up here, where it's M and P, we call it heterozygous. So that's a heterozygous state. This is a homozygous state. Is this possible? Could it become M and M from these parents? Well, yeah, look, the sire was KM, so there's an M, and the other M's come over here. They were both actually heterozygous, but both matching. They were both KM at that particular marker. And if we work our way down, you'll actually find out that this parentage was proven. Okay, this was genetically possible. So that's how parentage testing or parentage verification works. We take a hair sample from the foal. We've already got on record in a database the 2 parents, and we can then compare it. So that's where parentage is verified. These are Cleveland Bay animals, by the way, and all I've done is I've anonymized the data. There's a very good reason for anonymizing this one, because this is a case where actually the parentage isn't proved. So it's the same markers. This is a different mating. They've applied to register this animal. The hares have gone away to the lab, and the results have come back and said, it's not what you think. Let's just have a quick look. Same sort of table, different parents. Progeny tested at the first marker, NP. Well, N could have come from the sire, but it could equally have come from the dam. But where the heck did P, anywhere. Where did P come from? KM, is that possible? Well, yep, K could have come from the dam, M could have come from the sire. JJ, well, there's a J over here with the sire, but the dam isn't carrying J at all. It's homozygous for H, so that isn't possible. LN, is that possible? Well, yep, there's an L and there's an N. That was possible. There's a gap here, and the reason is that early on in the testing, not all the markers were tested, or perhaps they've tested and the results weren't very conclusive, so they've left them blank at that marker. But if we work our way down, some of these are possible, but some are definitely not possible. And so this parentage wasn't proven. They had to go away and go, actually, it was a young colt that had jumped over the hedge that had covered this mare, not the stallion we thought. And the reason I've been going through this parentage testing and showing you this is really just to lead up to this one, which is a parentage testing result of highly inbred mating. And I mean very highly inbred mating. This is actually more inbred than a full brother, full sister mating. I'll talk about how we measure that later. But this is a genuine result. Again, I've anonymized it. I will say that both the parents and this progeny are now deceased. So if there are any Cleveland Bay people in the audience, this isn't a living animal in any shape or form. This is historic, but this is a classic example of what goes on when we do highly, or can go on when we do highly inbred matings. So this is for real. And what I want you to have a quick look at is, oh, the parentage here is proven, by the way, that's not in question. But just have a look and see how many markers are homozygous. The sire was homozygous at 1, 2, 3, 4, 5 markers. Okay, 5 markers. The dam was homozygous. That's not atypical. In Cleveland Bays, it's not unusual to be homozygous at 5 markers. The dam was homozygous at 1. Again, there's one there that wasn't properly tested. Okay, we won't count that. So it was 1, 2, 3 markers. So 5 homozygous at 5, homozygous at 3. Now let's have a look at the foal. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Out of the 17 markers, that foal is homozygous at 11. That's a huge shift, right? The amount of genetic diversity that's been lost is incredible. And the amount of homozygosity that we're building in the next generation is incredible. It's just interesting to see it in that form. Okay, so I've talked about inbreeding, but what do we actually mean by inbreeding? And basically, it's how related an animal is to itself. That might sound crazy, but it's how related it is to itself down pathways that you can trace from the father up through the pedigree to the progeny, and then back down the pedigree to the founders on the dam side. So how many common pathways are there? If you want a scientific definition, don't write it down, don't even bother trying to remember it. We can calculate what we call an inbreeding coefficient, and that's the probability that any randomly chosen neutral locus, 2 alleles carried by an individual, are what they call identical by descent. That's definition's been around since 1922. There's a botanist in America called Sewell Wright. He came up with the concept of inbreeding coefficient. So let's have a look at it in the form of our original diagram. We've got our dam and our sire that are completely unrelated. And if we do those crosses that we did before, these animals, they're not inbred. They were the progeny of unrelated parents. But if we take this brother and sister and we cross them, then we've got inbred progeny at quite a high level. In fact, a brother-sister mating would give us inbreeding of 25%. That's what a full brother, full sister mating is. It's also the same for a sire and his own female progeny, or a dam and her own male progeny will give us inbreeding of 25%, assuming that the sire and the dam were unrelated in the first place. And of course, that actually is really true within our closed populations. So we can get inbreeding considerably higher than that 25%. And I'll show you that later. But I've shown you how inbreeding can reduce genetic diversity. What that in turn can do is it reduces biological fitness. I don't mean how well our horse can jump over a 5-bar gate. I mean how easy it is to get in foal, how well those foals go on to do, do they survive to full term and do they thrive? What it also does as inbreeding increases is it greatly increases the probability of what we call these deleterious alleles, these genetically damaging traits being expressed. And in a paper that I read recently, there could be as many as 46,000 potential deleterious alleles now detected in the equine genome. By the equine genome, what I mean is, the whole map of the 2.4 billion genes that makes up a horse. There's now research that shows there could be as many as 46,000 of those genes that are potentially damaging, and that's an awful lot more than we originally, we were originally aware of. And I've shown you some very shallow inbreeding in those diagrams, but we can, of course, have very, very deep inbreeding that actually brings together founders that, from many generations back in a pedigree. And with the digitization of the Cleveland Bay stud book, we're no longer looking at a pedigree drawn out on a piece of A4 paper. We're looking at pedigrees that are 10, 20, 30 plus 36 generations for an awful lot of Cleveland Bays now. There's over 360 years of breeding. So to put this in real context, and certainly into the Cleveland Bay context, I digitized our stud book in the mid-1990s and started to analyze it. And if we look at the pattern of inbreeding that revealed that went on during the last century, between 1900 and the millennium, at the start of the 1900s, inbreeding was down at around 0.02, 03, 04. It was quite low. It slowly increased during the Great War years, and then it suddenly decreased. Well, I can actually tell you that within a genuine closed population isn't possible. And the reason for that decrease was the amalgamation of two stud books. There was the Cleveland Bay Horse Society stud book that started in 1885, but there was also a Yorkshire Coach Horse stud book that existed in the very early years of the 1900s. And it existed for what were essentially part-bred Cleveland, first generation part-bred Cleveland Bays, because they found out that if you crossed the purebred Cleveland Bay with the thoroughbred, and in the late 1800s, we had thoroughbred horses, and the early thoroughbred founders were up there in northeast England, and if you crossed them with Cleveland Bay mares, you got driving horses that could pull the coach from York to London in less than 24 hours. And they were very highly desirable. And that was one nail in the coffin of the purebred Cleveland Bay. But that stud book only existed for 3 volumes, and the Yorkshire Coach Horse Society folded, but they amalgamated the stud books, which is why we got this sudden decrease in the immediate post-war years. It came down effectively to 0 by about 1935. But since then, look what's happened. It's increased in a pretty linear way, such that by the time you get through to 1990 and approaching the millennium, we're up here at over 20%, 0.2. And if I tell you that 0.125 is a full cousin mating, And there's a really good reason we don't marry our cousins. Okay, that rings alarm bells. Just looking at other equine breeds to see where we're at. I know we've got friends from the Suffolk Horse Society, and their stud book is just as old as the Cleveland Bays, but it's not digitized back as far. It's digitized back to the 1930s. We know that from the digital record, inbreeding in Suffolks is around and about 0.125%. It's at full cousin level. Just looking at some others, inbreeding here is measured in two different ways. We've got it measured for pedigree on the left-hand side of the table, but we've also got it measured in genetic means from looking at how homozygous an animal is, actually looking at the markers and seeing how homozygous animals are. And if we look at horses like the Friesians are known to be highly inbred, amongst the most highly inbred of equines. If we look at pedigree, for this particular population, this wasn't a global population, this was a Scandinavian population, they're up at 25%. If you look at it in molecular terms, it was 22%. Fairly close correlation there. I actually pulled this data together when I was doing a talk for Shetland Pony Society in Scandinavia, and they were up at about 17%, with molecular at 14, just trailing behind it. But there's a fairly typical snapshot across a number of equine breeds. There's the reference there, by the way, if you want to follow that one up. It is an open access journal. You can access that data quite easily on the internet. Let's look at where we're at with Dale's ponies. And this data comes from a 2017 analysis of the grassroots data that was presented to Rare Breed Survival Trust for analysis. And what it shows us is a fairly similar sort of pattern in that, albeit later than the 1930s, it's certainly post-World War II, inbreeding is the purple line, by the way. The green one is average genetic relatedness, which is it's closely mirrors it. It's not quite the same thing. It's the purple line that we're interested in. And if we have a look, certainly from the 1950s, it's been increasing in a pretty linear way up through to about the millennium. There's something that's gone on here that's made it decrease slightly, but then look, there's big signs of it starting to increase again. But that linear profile's there. It's certainly not getting up to 20% like in Cleveland Bays, but in 2017, it's certainly gone past the full cousin level, and it's approaching 13%, I think. If we look at it in tabular form, this was the pattern over the millennium through to 2014, and this is, sorry, I'll just go back one. This is minimum inbreeding, maximum inbreeding, and the average, and this is the standard deviation on that. This is the number of animals being foaled or being registered. And if we look here in the most recent, we've got 2017 here. The minimum inbreeding was down at 0.05, but the maximum inbreeding was up at over 40%. Now, how can that be when a full brother sister mating is only 25%? Well, if you already start with inbred parents, we will only increase inbreeding if we bring them together. The average for the breed in 2017, 0.14, so 14%. And if you look down that column, you can see that the trend has been for, yes, there are lumps and bumps along the way, but the general trend is for it to be increasing. This is a picture of inbreeding in some of those other breeds, but looked at from a molecular point of view. We've got the Friesian horse there as the most inbred, Shetland pony closely following it. When we look at runs of homozygosity, ROH, runs of homozygosity within the genetic makeup of an animal, we get these chains of alleles all next to each other that are homozygous, and the length of those chains can give us an indication of whether that inbreeding is very recent in the pedigree, whether it's medium deep in the pedigree, or whether it's very, very deep in the pedigree. It depends on the length of the chain. And what does it, does this really matter? Well, this is something called the extinction vortex, and it's something we need to be aware of, because if we start with a small population and that population is a closed population, So whether we like it or not, there is going to be inbreeding going on, and we're going to be losing genetics by random genetic drift. Well, we'll lose variability. We will carry on round the spiral. We lose genetic fitness and the ability of the population to adapt to change. We've got lower reproductive rate. We've got higher mortality, which leads to an even smaller population. So this spiral exists, and what we need to do is to try and uncoil that spiral, or better still, step off of it. But we need to be aware of it, because this extinction vortex, if we go too far around that spiral, we actually end up with the whole population going right down the plug hole. I want to talk a little bit about inbreeding depression, but more particularly about deleterious alleles, because it is very relevant for the Dale's pony, and we'll look at why in a moment. But let's suppose instead of A, B, C, and D, like our sire and our dam were carrying originally, unrelated animals, let's suppose our dam is still carrying A and B, but this time our sire is carrying C and X. And X is a rare but deleterious damaging gene, okay, a damaging allele. And if we do our cross, this filly foal is now carrying AX, and the colt foal is carrying BX. They've both inherited X from the father. Now, if it's what we call a deleterious recessive allele, X is recessive, it's masked by a more dominant gene. So this sire won't actually be showing this problem in its makeup, in the way it appears, nor will the problem be manifest. But if you inherit two copies, you will. And if we cross these two carriers now, in this filly foal is XB. So it is a carrier, but it won't actually be exhibiting the problem. But this colt foal will. It's got two copies, and so it's expressed it. And if we look at some of the traits that we know are deleterious in pony breeds, in Shetland ponies, for example, they've got problems with dwarfism and what we call skeletal atavism. It's prevalent in miniature horses as well as Shetland ponies. So in miniaturism, we get these horses with tiny bodies and big heads, but we also get, you know, this excessive distortion of the limbs. In particular, if you look at, I mean, I would call that cow hock, but it's beyond that. Those of you who know anything about Connemara ponies, it's not exclusive to Connemara's, but Connemara's have got a problem with what they call hoof wall separation. That again is a deleterious recessive allele that expresses itself when you get two carriers are mated, and it gets expressed. Anyone that's involved with quarter horses will know about polysaccharide storage myopathy, shivers, where you've got this really tucked up horse at the back and a big horse at the front. But this is the one that is of particular interest to Dale's ponies and to fell ponies. It's almost certainly something, a mutation that occurred within the fell pony breed, and it's actually crossed through admixture into Dale's ponies as well, foal immunodeficiency syndrome. And I know that in the Dale's pony world, there's a very rigid testing for stallions up for licensing, and you do not allow stallions that are carriers to be granted a license for very good reason. Just looking at how this foal immunodeficiency is passed on, if we've got let's have a look at the situation on the right, where we've got one carrier in the mix here. So we've got a female that isn't carrying it at all, and we've got a male that is carrying it. Okay, so we've got the ova at the top, we've got the sperm down the side, and if we cross those, this is basic Mendelian genetics. You might remember it from biology lessons and sweet peas. If we do that cross, what are the probabilities of that gene being passed on? Well, if we do the cross, we've got NN not carrying it, NN not carrying it, NFIS, so we've got a carrier and we've got another carrier. So in other words, if we do that cross of a carrier with a non-carrier, we end up with the chance of 50% of the progeny being clear and 50% of the progeny being carriers. You will not get it expressed on the ground because it's impossible for it to have two copies of the damaging gene. But if both animals are carriers, so the dam was carrying it and the sire was carrying it, the proportions change completely. So in this case, we've got 25% are clear. We've got 50%, there's a one here and another one here, that are carriers. But the red one has inherited two copies of the damaging gene and it's expressed. And in the case of foal immunodeficiency syndrome, the chances of that foal surviving through and being brought up and living a mature life is very, very low, as you know. And it's the same if we've got what we call autosomal recessive genes. In other words, the damaging trait is recessive. Then if we mate two animals that don't carry it, they will be 100% clear. If we mate one that's clear and one that's a carrier, as I said before, it's 50-50. If we mate a carrier with somebody that's clear, we get 100% carriers. No, sorry, with an animal that's affected. If you could mate one with us affected, you'll get 100% carriers. And if we look at it from the other end, if we make two affected animals, they're obviously both going to be affected. This is the situation where you make two carriers, 25% clear, 25% affected, and 50% carriers. So that's total justification for making sure that if you license stallions, they're not carriers. I know we've got friends from the Exmoor Pony Society in, and I wanted to mention this because it is possible for animals to survive and even to thrive through a process of natural purging, because these deleterious mutations, we know about mutations from COVID now, don't we? The Omicron variant and things, and Delta. As these mutations happen naturally, not only do they occur, but they also get purged naturally. And within some populations that tend to be left to get on with it in a random way, it is possible to survive and to thrive. These are Chillingham cattle. They're probably amongst the rarest of cattle breeds in Britain, if not the world. They are almost clones of each other. They're genetically very, very homozygous. To use the word cloning is wrong. I can get pulled up for that. It's not Dolly the sheep. But they are very homozygous and they're they're almost genetically identical. But yet they're so highly inbred, but they survive. When I did my PhD, one of my supervisors was Professor Stephen Hall, and he's the bloke in the world that knows more about Chillingham cattle than anybody else. And I said, well, why does this process of natural purging happen? And why can't we just manage all other livestock breeds as if they were Chillingham cattle, and deleterious alleles won't be a problem anymore? And he says, it's not as simple as that. And, really the chances of managing a population in a highly inbred way, continuing to inbreed on purpose, is probably not a strategy that you can bank on unless you really know what you're doing. And I'm guarding my words here because there are a very small cluster of very knowledgeable people within the Exmoor world who are managing to maintain very small subpopulations within restricted, isolated herds, knowing what they are doing, and are managing to avoid the problems of accumulating deleterious alleles. But it's a very specialized situation, and it's not something that I would at all recommend a breed society or individual breeders to adopt. So it is possible to survive in a very inbred state, but it's not the norm. And part of the reason it's not the norm is because we tend to intervene in our livestock breeding. We don't let animals randomly mate. We choose what dam we breed from, and we choose the sire very carefully. And we tend to call the vet when things go wrong. We don't just let foals, get aborted behind the hedge and do nothing about it. And we certainly, we tend to call the vet when things are starting to go wrong. So, Chillingham cattle run in a very feral way on a restricted parkland up there in the northeast. And if the vet's called, then it's a case of darting the animals and treating them like captive wildlife more than anything. Okay, so we talked a lot about inbreeding, about the relatedness of an animal to itself through its sire and its dam. What I want to talk about now is about kinship and the subtle difference between kinship and inbreeding. And just as inbreeding is related in the relatedness of myself to myself through my mother and my father, if they've got a common ancestor, kinship is the relatedness of any one animal to any other animal. It's how related I am in the human UK population to any one of the participants within this meeting through an ancestor that we share that perhaps we weren't aware of. So just as we can quantify inbreeding, we can quantify the relatedness of any two individuals in a population with what we call the kinship coefficient. And that has also got a biological definition that is to do with identity by descent down through the pathways, and it's pathways of a pedigree from one individual to another sharing a common ancestor, that two randomly chosen neutral alleles from the same locus in two individuals are identical by descent. Now, full siblings are 50% related. In other words, they've got a kinship coefficient of 0.5. If you remember, I said full siblings were 25, potentially, full siblings that are mated, are 25% inbred. Okay, so kinship can be double the inbreeding. If we look at that, we make these two animals that are not related. We've already said an F, by the way, is the biological symbol for inbreeding coefficient. They're not inbred in this next generation, but they are related. They share common parents. So the kinship coefficient is 0.5. They're 50% related through these two parents. If we mate these two animals in this generation, we've already said they were going to be 25% inbred. Their kinship increases by 25% to become 75%. Okay, and that process continues. If we make these two, inbreeding coefficient goes up to 37%. Remember, I said we can be more inbred than that 25% of a full brother, full sister mating. That's how it can happen. These chains of inbreeding, and similarly, the kinship coefficient will increase as well. If we were to take a population of horses, ponies, human beings, and we looked at the kinship coefficient between every individual and we averaged it out, we would be able to come up with what we call a mean kinship for the whole population. And mean kinship is exactly that, the average of all the kinship coefficients between all the individuals and all the living, that's the important bit, all living individuals in a population. And it quantifies the relationship of any one individual to the rest of the population, the rest of the living population. So it's a dynamic thing. It's going to change as the population changes, as animals are foaled, as animals die, and as colts are gelded as well, actually, and can no longer contribute to future generations if they aren't collected before they're cut. It is an indication of genetic diversity, the genetic health of a population. It's not the best measure, but it is a measure. And it also gives us an indication of how genetically important an individual is within that population. Now, I know we're managing rare breeds and all of the animals are important, but in a bit of an animal farm way, some are more important than others because they're carrying the genes that are more at risk of being lost. If an animal has a very low mean kinship within a population, it is carrying rarer genetics and those genetics will be at greater risk of being lost through genetic drift. So that actually gives us an indication of animals that perhaps we ought to prioritize for gene banking, for semen collection and things like that. Okay, so I said at the beginning in the overview, we talk about inbreeding, we talk about kinship, but I also wanted to talk about something called effective population size. And what do we actually understand by effective population size? Is it just population size? Is it the number of who's we've got on the ground? I think what we need to do is we need to understand the difference between an idealized population and a real world population. And in biological terms, idealized populations, they are infinitely big. They are subject to random mating, like Chillingham cattle. And all individuals, be they male or female, get the chance to contribute equally to the next generation. And it's all nice and tidy, and generations don't overlap. And if you look at any one of those four things, none of them relate to the real world livestock breeding. Because in real populations, you know, we can't afford the luxury of an infinitely large population. Livestock breeding, be that horses, cattle, sheep, or whatever, we maintain skewed populations. We keep far more breeding females than we do breeding males, certainly in equine breeding. We have unequal family sizes. And we as human beings make selection choices. You know, we choose what dams we breed from and what sires we use. You know, it is not random mating. It's far from it. And of course, any of you that have tried to unravel a pedigree will realize that our equine stud books are just enmeshed in overlapping generations. It's not all neat and tidy. So to be able to actually measure a population, we come up with this concept of an effective population size, and that is using the data from our real world population. How does that equate to it if it was translated into the size of an ideal population? And it is one of the most important parameters in population genetics and conservation biology. It translates real census size, if we go around and count up our horses and ponies, of a real population into the size of an idealized population that is losing genetic diversity at the same rate as the population that we're concerned with. And NE is the symbol, the biological symbol for effective population size, and it tends to get measured in two different ways. It can be measured from the rate of increase in inbreeding, and it can be measured from the number of parent animals that are contributing to the next generation, from the number of sires we're breeding from, and from the number of females. And any of you that were aware of the previous watch list, it was based on the number of breeding females. So it was skewed, and it wasn't taking any account of the contribution of males to the population. And that's why the system has recently changed. And in fact, what they're doing is they're measuring from census effective population size, because that's what they can get reliable data from breed societies. But in fact, that in itself is a bit of a skew, because the only real way to measure effective population size from pedigree is to measure it from the rate of increase in inbreeding. And if you want to chase that one back, the person that stressed that was John Williams from the Roslin Institute. And you know, it's really important we bear that in mind when we were reporting to the Food and Agricultural Organization of the United Nations, because that's what they actually want to know. And in fact, government is well aware of the limitations of what it's doing at the moment. There is a concept called the 5500 rule, which tells us that if we measure effective population size from inbreeding, we want our populations to be greater than an effective population size of 50 to be able to survive in the short term. And we want populations to have an effective population size of over 500, albeit measured from census effective population size, to survive in the long term. It's something called the 5500 rule. So we need more than 50 to survive short term, more than 500 to be long-term viable. In fact, some geneticists are saying that we need to double that. It needs to be 100, 1,000, but I'm not getting dragged into that debate at the moment. But that's the bit, at least 50 for short-term survival. And in fact, the figure of 50 is a bar that is recognized by the United Nations, because with an effective population size of 50, a population is losing 1% of its genetic diversity per generation, not per year, per generation. And with most equines, certainly with Cleveland Bays, I'm pretty sure Dale's Ponies mirrors it. The generation interval is 9 years. So every 9 years, you're losing no more than 1% of your genetic diversity. And we need more than 500 to survive long term. If you want to chase up the reference for that, that's from Franklin 1980. So that's been around for a long time, the 5,500 rule. It's all well and good having these rules and the data, but how do we study the data? There are a number of ways, but there is an open source piece of software called PopRep that was put together in Germany for the Food and Agriculture Organization of the United Nations, actually. And that is what the Rare Breed Survival Trust uses. And the figures that I'm quoting for Dale's Ponies and for Cleveland Bays tonight are based on what we call a PopRep analysis of the stud book. The figures that I've got for Dale's Ponies are from a 2017 PopRep analysis. The ones from Cleveland Bays are from 2022 analysis. If we look at Dale's Ponies and that 2017 analysis, where is the Dale's Pony population? There are a number of different subgroups of measuring from the rate of increase in inbreeding, but from census population size, what we can actually see is that that's the purple line. We've got a population that in 1990, it was of the order of 200. It's now probably around about 300, getting towards the 400, slightly increasing. But remember that census effective population size from the number of breeding males and females, it isn't at the 500. And I'll pop over to the next chart, which makes it easier to understand, but each one of these is a different method of measuring from inbreeding. If we have a look, this is our bar of 50. There's some figures that are bouncing around all over the place and are unreliable. But if we look at the most reliable method, PopRep actually looks at all the different methods based on the data that that is available and looks at the most stable of the different methods. And what it's suggesting here is that the figure that's most stable and reliable for the Dale's population, taking that 2017 data, was that in 2017, the effective population size measured the proper way was 63. Well, that's the right side of 50. That's the good news. But what I would show is the trend, because in 2012, it was 76. Yes, it's bounced about a bit, but over that 5-year period, the trend is for it to be decreasing. So whilst it's above that red line in the sand, it is moving towards it, or certainly it was during that period 2012 to 2017. To put things in perspective, let's have a quick look at some other breeds. Again, we've got it measured from pedigree-based pedigree, but we've also got it measured genetically from the runs of homozygosity. If we have a quick look at Shetlands, for example, which is the one that I was particularly interested in before, the Shetland pony population, 365 there from census effective population size. Okay, if we look at maximum and minimum inbreeding there in the Shetlands, we've already seen we've got minimum inbreeding there of 10%. We've got a maximum inbreeding there of 37% in this particular population and 18%. So just running down that table, we've got a lot of populations there that are between 250. Not one of those is over 500. So perhaps, you know, it's a problem that's actually more widespread than is recognized across equine populations, that effective population sizes are fairly compromised. Just looking at what can happen and looking at loss of variation, if we have an effective population size of 100, we're losing 0.5% of our variation per generation. I said that 50 had been set as a line in the sand because it was 1% per generation. If we halve it, we double. So if we halve our effective population size to 25, we double the loss to 2%. Halve it again, it's been rounded off there, but we get to 4%. And if we halve it again, get down to an effective population size of 6, we're losing over 8% of genetic variation in a generation. I know that at the start of the 1990s, Cleveland Bay effective population size was below 20. So they were getting on, they were certainly losing more than 2%, getting towards 4% per generation. That was what was being lost. The other thing that I mentioned before, with idealized populations, we try to balance out and get as much contribution from males and females as possible. But I also said that in the real world, we maintain our equine populations in a very skewed way. If we were to maintain a population of 100 with 50 females and 50 breeding males, effective population size of 100, just as in the last table, we're losing 0.5% per generation. But if we still maintain the population at 100, look at what happens to effective population size if we skew it so that we've got 25 breeding males and 75 breeding females. Effective population size is now down to 75 and we're losing 0.7%. If we skew it even more to 10 and 90, we've still got 100 horses. It's getting closer to how we maintain our equine populations. We've now got 36 is our effective population size, and we're losing 1.4% per generation. Let's get down to something even more realistic. If we get down to 2 stallions and 98 females, effective population size is only 8, and we're losing 6.5% per generation. And that's not atypical of an equine population. So with our breeds, we've got, choices. How do we manage them? Well, one strategy, like they did with zoo animals, is to divide the populations up, send them off to zoos around the world, and just keep them completely separate and let them develop on their own. So different populations on different sides of the Atlantic could develop separately. They will develop their own mutations, and that will actually help maintain diversity. We could do the Chilean cattle thing and just let animals randomly mate, but there are very good reasons we don't do that. So the gold standard, and I'll say that with a caveat, method of maintaining populations, be they zoo animals or rare equine populations, is to manage them by managing inbreeding. But we manage inbreeding not directly. And what we do is we manage it by, first of all, considering mean kinship, and then, in other words, the relatedness of the animals that we bring together, and then the potential inbreeding coefficient of the progeny. And I don't know if any of you folks are involved in canine breeding, but the Kennel Club have invested huge amount of money in a very sophisticated piece of software that enables breeders of Shetland sheepdogs, Jack Russells, or whatever it is, to look at the theoretical inbreeding coefficient of any potential mating, any pair within their pedigree. And it tells you the lowest inbreeding coefficient to try to bring inbreeding down, because they're aware that inbreeding is a problem in not just equine breeding, but in canine breeding as well. The problem is that what they haven't done is taken the lessons that have been learned from captive wildlife breeding, where they've done all the computer modeling and they've worked with animals that have got a much shorter generation interval in the real world. And they know that if you just have a minimum inbreeding strategy and you do it directly, what happens is, yes, you get a reduction in breeding in generation 1. In generation 2, you might see that reduction as well if you carry on the strategy. But by the time you get to the 3rd generation, you're running out of places to go for a low inbreeding mating. And by 4 generations down the road, you've brought everything together. You've mixed the rarest genetics with the most common genetics in your breed. You can't separate them out again. And you actually get a rebound. And then suddenly inbreeding increases at a greater rate than it was before. So in other words, shoot yourself in the foot. And the Kennel Club canine average generation interval is a lot less than in equines, they're going to run into a problem very, very quickly if they don't address that. And it's not just Andy Dell saying that. There's canine breeders outside the UK that have been saying that for quite some time. So what will minimizing mean kinship do? It will reduce the rate of increase in inbreeding. It will stop us mixing together all of our genetics are rare, but it would stop us mixing together the rarest of the rare and the more common of the rare. And the problem is, if you do that, if you mix them together, not only do you increase the frequency of the rare genetics that you really want to maintain, but you're also increasing the frequency of the more common genetics as well. You can't separate them out very easily. It helps us to maintain as much genetic diversity as we can. In other words, what it does is it slows down the amount of genetic diversity that's going down that plug hole of the vortex. It turns the tap down. We'll never stop it completely, but it can turn it down to a drip. And it also, minimizing inbreeding, the awareness of mean kinship helps us identify genetically important animals in our population and prioritize breeders, whether that's actually just for encouraging owners of the rarer mares to actually continue continue actively breeding, or for example, if we've got a colt that's born that's very low mean kinship and it meets breed standards and there's no other reason for not doing, if it can't be maintained entire and run on to have a useful life as a stallion in itself, we can use some of the more modern technologies to actually collect the testes at the time that it's gelded, get them off to a lab and get the semen collected and frozen down. All of what I've talked about, yes, I've hinted at DNA and measuring inbreeding genomically, but all of the stuff that I've talked about has been focused on pedigree. And pedigree analysis, what it does is it gives us a whole load of probabilities. But that's it. That's the limitation. It's just probability. And in an era where genomics, our ability to do way more than just the parentage testing verification, that's called, looking at 17 markers out of the 2.4 billion genes in a horse's makeup. We're moving rapidly to be able to do what they call SNPs testing, where we're looking at 50,000, 70,000, 670,000 markers in a horse's genome, or do whole genomic study, get the whole genomic profile. How relevant is pedigree? Well, there's been studies on thoroughbreds in the USA and in Australia, particularly in Australia, that have compared inbreeding based on pedigree analysis and on genetic analysis. And what they have found out is that when they look at inbreeding based on pedigree, in pedigrees that are really deep, and we're talking with thoroughbreds just like Cleveland Bay's 30 to 36 generations deep now, there's actually a very, very good correlation between inbreeding assessed from runs of homozygosity with that was assessed from pedigree. And we saw it in the table that I showed earlier with the Friesians and the Shetlands. There's actually quite a good correlation. One tends to mirror the other. And as a breed society, your stud book data is a really valuable tool. And, there are pedigree-based methods. You haven't got to go away and suddenly do genetic testing, thorough genetic testing on every animal that you've got to be able to work out its pedigree-based inbreeding before you can think about how you manage the population. Okie doke. So with Cleveland Bays, with all this knowledge, what did we do? Well, first and foremost, I'm a horseman before I'm a geneticist. And I like to think of myself as a horse breeder with a biologist's hat on. And I like to think of myself as having a toolbox. And first and foremost in my toolbox is the horseman's eye. When I look at a horse and decide whether I'm going to breed from it, I want to know, has it got a leg at each corner? How does that horse move? Does it meet breed standard? And all those things. And from a genetics point of view, we can cluster that knowledge and term it under this umbrella of what we call phenotype, the physical makeup of the horse. But we've also got things about genotype, the genetic makeup of the horse. So those are the two things that we need to consider. And when I first started doing that with Cleveland Bays and trying to look at pedigree and trying to take on the genetic aspect of things, I probably, like yourselves, just got stud books out off the shelf and start drawing out pedigrees. I'm talking about the 1990s, before we had Geniped and PediWeb and could rapidly generate those pedigrees on screen. But even with generating pedigrees on screen, there are limitations. And I remember sitting down with a piece of A4 paper. Yes, you can do a 5-generation pedigree on A4. It's not a huge problem. Fits quite nicely. Gets a bit more difficult when you get to 8 generations. You tend to need to get sellotape and scissors out and start sticking bits of paper together, or you grab hold of a piece of A3 paper. Then you can draw it out. You can get it up on a computer screen relatively easily if you've got your glasses on. If you want to go deeper than that and go to 10 generations, suddenly we're not talking about the 32 animals that are at the base of a 5-generation pedigree, or the 256 animals at the base of a 8-generation pedigree. We're now talking about 1,000 animals on the base of that pedigree 10 generations back. You've got an awful lot of pieces of A4 paper to stick together. You've probably come off the kitchen table, you've gone onto the kitchen floor, and depending on the size of the kitchen, you've probably gone out on the patio on a windless day to lay those pieces of paper out. If you carry that process on and you go back 20 generations, there's absolutely no way you're doing that on your patio. And that's not just because of the wind. It's because we've gone a further 10 generations, but instead of having just over 1,000 animals at that baseline, we've now got over 1 million animals in that baseline. We're going to Wembley Stadium to lay out the pieces of A4 paper. So you can see why it isn't practical to go back 20 generations and fully understand inbreeding at that depth without the use of computers. You can't do it. I said that Cleveland Bays, you can easily go back 30 generations. 32 generations isn't a problem. For a lot of Cleveland Bay horses, you can go back 36 generations now. You can do that with thoroughbreds as well. And if you go back that far, you've got over 4 billion, not million, billion animals in that baseline. Now, hang on a minute. Current estimate of the global equine population is 58 million. That comes in between 25 and 26 generations. If all of those animals were completely unrelated and we lined them up, you could have a population 25 generations deep. But if we go any deeper than that, then you bet your bottom dollar there's inbreeding, because some of those founders had to be related. There aren't enough horses in the world. So inbreeding is inherent in equine populations. You know, they fall out beyond the beginning of any stud book. They were related. There's been big studies now of both mitochondrial DNA, which tells us about the female origins of equines, never mind equine breeds, but domestic equines themselves. I know, looking at Y chromosome studies to see where were horses first domesticated, probably on the plains of the Botai about 5,000 years ago. And man moved from the Botai, they moved east and west, taking their domesticated mares with them. How do they know they took domesticated mares with them? Well, because they've looked at pottery, and then they found lipids that come from milk within stoneware urns. And wild mare's milk doesn't end up in stoneware urns. They must have been milking them, and they've radiocarbon dated it back to that. And they think that man actually domesticated the female equine first, and potentially the early equines mated with wild males as they migrated east and west and came through towards Iberia and across towards what we now know as China. So a global population of 58 million nowadays, there's no way that our equine populations can't be inbred. And it is actually quite important that we do look back further than just our stud books. This is from some canine data, but it shows out quite nicely. I'll give you the link to this website later, because it's from a website called breedingfordiversity.com. And he quite cleverly shows what a population map looks like if you go back just 7 generations, but how rapidly that structure can change. If you incorporate all the generations, you get a totally different map of the structure of a population. So it is important that we actually go back as far as we can, certainly if we're looking at pedigree-based information, to try and do these analyses. So armed with all the data we managed to generate from computers and adopting software from the zoo world, called Spark, Single Population Animal Record Keeping System. I tweaked it and used the science behind it, but turned it into a more user-friendly piece of information that was of use originally to myself and a cluster of friends, but eventually to the whole breed society, and also beyond that to anyone that had an interest in breeding Cleveland Bay horses. It's now been extended to Eriskay ponies and to Suffolk horses, and there are another of other equine breeds. There's also other types of livestock that have adopted very, very similar systems. And what I want to do is just to go through that system and see how it works. And what it's evolved into are these mare-based tables. So there is a set of tables for every female of breeding age within the population, and it looks at the potential co-ancestry coefficient, the theoretical inbreeding of every cross with every potential licensed stallion. But it also looks at the mean kinship of the two potential parent animals, and it tries to identify animals of very similar mean kinship that can be mated together to produce animals of inbreeding coefficient of lower than the average for the breed. That's it's been refined a bit more than that, but that's the basic principle. So we've evolved that into a traffic light scheme, and if breeders follow those traffic lights, it still leaves breeders with a choice. You can see on this particular sheet there are a lot of green matings. They're highly desirable matings. We've got yellow matings. They're less desirable, but you know, they're best of the rest. If you can't do a green one, do a yellow one. There are orange matings which bring together animals of very different mean kinship. We'd rather you didn't do them. And then there are red matings, which are highly inbred matings. So the green matings, these are the ones that we're trying to encourage. They're matings of animals of very similar mean kinship. We've actually had to tweak this for Cleveland Bays, because if you just go on the average inbreeding coefficient for the breed, you get very few green matings. But we've actually determined that the best way to do it is by matings with progeny of lower co-ancestry coefficient than the mean kinship of the mare. That gives as many green matings as we could potentially get. It's close to mating to the average inbreeding for the breed, but slightly tweaked to make it more effective on the ground. Matings giving progeny lower than that average for the breed. The best of the rest is if people can't do that for whatever reason, you know, there aren't any stallions anywhere close, I don't want to use AI and bring in frozen semen, then the best of the rest matings. These are still matings of animals of similar mean kinship. We're keeping the animals at the two different ends of that mean kinship spectrum apart. We're going to get slightly higher co-ancestry. In other words, the inbreeding is a bit higher, but it's still below a threshold that we set for the breed. And those are the preferred matings if we can't do a green one. Then we've got these matings that we try to discourage because they are mating animals at the opposite ends of the spectrum. They're bringing together the blue Smarties and the red Smarties. They're producing matings of higher than the average co-ancestry as well. But still less than that red line that we draw in the sand of the breed threshold. And, you know, what these matings do is they do bring together those rare and the common genetics and mix them together. So we're trying to get people to avoid those if we can. And then there are these ones that we really want avoiding. These are the ones like that parentage testing that showed that rapid increase in homozygosity. These are extremely different mean kinship, but they're also very highly inbred. And for Cleveland Bays, that's greater than 24%. Our average inbreeding already at 22%. 24% pitched just below that 25% brother-sister threshold. For Eriskay ponies, it's at 18%. For Suffolk horses, it's at 0.125 at full cousin mating level. Progeny from those matings are highly inbred, and they will, if we look at the pattern of inheritance of deleterious traits, it increases the chance of deleterious traits being expressed in the future. So since the implementation of that scheme within Cleveland Bays, it unofficially became available mid-1990s, but was officially implemented in 2004. What's actually happened, I already showed you that graph of increasing inbreeding, but since 2004, certainly since about 1996, when I first started informally issuing the sheets, but 2004, where they were officially adopted by the society, and we've had best part of 16 years of the mating strategy now. You can see that inbreeding is still increasing, but it's increasing at a much slower rate, which is what we wanted to happen. And in turn, what that's actually done to effective population size, if we look at where effective population size was at the turn of the millennium, as I said, it was down at 20, way below that 50 threshold in the sand. Well, it bumped along, but after the official launch of the scheme, it steadily increased, such that by 2013-14, it went through the 50. It's carried on increasing. Yes, there's been some hiccups along the way, but by 2020, it had got to 125 off the top of my head. And now most recent figures show it's past 200. So we've actually managed to stem that increase in inbreeding. And through doing that, we've managed to increase effective population size. In other words, we've turned off that pouring tap of genetic diversity, and we've now reduced it to a drip. And we are managing to stem the loss of diversity. If you remember, at 50, we would be losing 1% per generation. We were losing more than that. We're now losing less than that. So the rate of increase in inbreeding is really important to monitor it and to slow it down. It will never become zero. The only way to bring it to zero is if you actually open your stud book up. If you remember what I showed you, the graph of when they amalgamated the two stud books, it decreased. Well, yes, if you bring animals in from outside, you can reduce inbreeding to zero, but that's not a good thing to do. We're trying to slow it down, but slow it down, not get it to turn around and start decreasing. And we want it to decrease slowly. All right, we want that we want to stem that tide, because if we do what the Kennel Club are doing and turn inbreeding backwards, there will be a rebound 2 or 3 generations down the road that we will come to regret. Because what the Kennel Club has done and what that strategy will do is it will cap the maximum reduction in the future. That's been proved from not just from computer modeling, but from the true world of captive wildlife management. And that's why we're managing it through mean kinship first and foremost, and not directly through inbreeding. In the real world, there are problems. Just as actually a distribution of Eriskay stallions around the country and Eriskay breeding mares, and it's, you know, it is quite hard for a very small population like the Eriskay pony. You know, geography does present problems. It presents huge problems, but there are ways around about it. You know, traveling distance, we can provide traveling grants, but we can also overcome that by starting to use artificial insemination and providing grants for using artificial insemination. You know, isolated herds, the Eriskay's got a particular problem. You know, they got island herds. As I say, traveling grants can help. The other problem is that skewing of the population, the fact that we maintain more breeding females than males, that actually not everybody has got the facility to run the colt on, you know, bolshy years between, you know, when they're a yearling and a 2-year-old, to actually to get them to a state where you could potentially use them as a stallion. Well, there are some, for the Highland ponies, for example, have a colt retention scheme that actually helps farm colts out onto facilities that can run them on until they become stallions. We've also got more modern technologies now. As things improve, then yeah, we've got semen collection, but we can also harvest from gonads, from testes at the point that a colt is gelded. If we can get the colt to still be entire at say 28 months old, from 28 months onwards, it's viable to collect viable semen from the testes, from the epididymal semen collection. So things to consider when we are selecting matings. We need to try to breed from as many males and females as possible, and some Suffolk breeders in amongst us will recognize Craig Howell Jensen there. Very much to the fore 3 or 4 years ago, he came into the hands of a very proactive Suffolk horse breeder. There's also a Cleveland Bay breeder, a friend of mine, who is very aware of the problems that it could potentially cause, but he's also very good at marketing his stallion. This horse effectively came out of the woodwork. It hadn't been overused, but suddenly he got to Stallion AI, and with a lot of publicity, he was collected, and he covered an awful lot of mares that year. And the following year's foal crop was very heavily skewed towards him. There were 11 foals on the ground by Jensen's in the following year. And whilst it's great to have foals on the ground, it has a knock-on effect if we skew it towards any one stallion. But what we're trying to do is to increase effective population size, and the sheer fact that 11 were by Jensen that year actually caused a dip in effective population size. And that's not a slighting of Bruce, because I think overall what he did with Jensen was really good, because out with the Suffolk Horse Society, he really raised the profile of heavy horses, and it brought in it helped RBST an awful lot with their heavy horse appeal and the amount of money that came in. So it did an awful lot of good. It also helped move Sparks on a little bit, in that we provide stallion-based data as well as mare-based data now. And there are other strategies that we can mitigate against that. But we do need to avoid the overuse of any small group of stallions. And, you know, from native ponies on a whole, I think Sparks, or a Sparks-like breed management strategy, is potentially another tool in the toolbox. The good thing about Sparks is it very much leaves breeders in control. It's not a breed society dictating how a breeder should breed. It's a little bit stick and carrot, certainly from the Cleveland Bay point of view, in that we use horse betting levy board money to provide incentives. We do incentivize green matings, and also we actually penalize red matings that we don't provide any foal grants for foals that we still have to register the animals, because zootech regulations say we have to. They have to go in the pure stud book. But what we can do is not give any foaling grants to highly inbred matings that are red on our Spark sheets. That discourages those highly damaging matings, and we still see one or two, but nothing like the number that we were seeing. What that does is it maximizes retention of diversity, and we can also encourage mare owners to breed from those low-banded mares. And actually, Cleveland Bay Horse Society ran a selective mating scheme where we tried to get as many green matings from those low-banded mares as possible. And we managed to get extra horse betting levy board money specifically for that program. So if we bring that into the Dales world, for the future of the Dales pony, I think that pop rep analysis, the Rare Breeds Survival Trust lasted one in 2017. I think it would be quite useful to do that again with another set of data, and I'm sure they will give via grassroots access to Rare Breeds Survival Trust. But rather than just fire the figures up to DEFRA, it's probably worth, you know, getting that looked at. Let's see, where is the breed in the 5 years since 2017? Is the trend still for effective population size to be decreasing? Has it leveled out? Is it increasing? Has it turned? Sparks, it's a possibility. I provide it free to other equine rare breeds. The more that come on board, the more problematic that comes, simply because everybody wants the information at the start of the breeding season. For Cleveland Bays, we've got horses around the world. It's one global population. For Suffolk horses, it's a UK, and for Eriskays, it's a UK population. Yeah, but a Sparks type breed management is possible. Horse betting levy board grants, I know the Suffolks are starting to target horse betting levy board money at breed management schemes. Cleveland Bays certainly are. Yeah, Rare Breed Survival Trust very much involved. They, like I say, are reporting back to DEFRA and to FANGA. I show this slide, which Suffolk breeders may recognise. It's Ruby and her filly foal Hope. Ruby went to Stallion AI and was the recipient of sex semen. Sex semen, relatively new technology in the equine world. It's been around in the cattle breeding world for a good few years, but it's been problematic with equines. But we were fortunate in actually being able to select a green mating. It was a Sparks fully compliant mating. It was the second stallion that had gone there for testing for sex semen, and we produced that foal hope for the breed. She went back to Brackenhurst to Nottingham Trent University, has now been sold on, and has gone back to the stud where Ruby was originally foaled. And so, you know, hopefully she will go on to become part of the active breeding population. So, you know, modern technology can help these breeds, but what is pretty clear is that uncoordinated breeding can lead to unsustainable accumulation of inbreeding that has a direct and a negative effect on effective population size. As I say, I think it is important that breeds do keep up with the pop rep analysis. Potentially, if that analysis reveals that there is a growing problem, they could look towards a Sparks type breed management strategy, or I'll flag up that there is another breed management system similar to Grassroots called Cloudlines that's been developed that has a similar system built into it. But above all, what you mustn't lose track of is EU Zootech regulation 2016 that's now become the UK Animal Breeding Regulations 2019. It's almost word for word the same legislation. And what that really does is it entrenches breeding decisions firmly in the hands of the individual breeder and not breed societies. So breed societies and breeders need to work together to come together for the common good of our breeds. I mentioned this website, Breeding for Diversity, a little while back in the canine world and looking at that population structure diagram. I'll just pop it up there again if you want to scribble that down, or obviously it will now appear in the recording of the meeting, www.breedingfordiversity.com. He talks about 4 steps for conservation of captive populations and also for the management of livestock populations. He goes back over the ground that I've gone, but into a bit more detail as well about the strategy of managing breeds through using mean kinship. I point you to that. It's a really good guide. In a layman's terms, he's very good at that. If you want a bit more about the Cleveland Bay project in itself, there's now a peer-reviewed paper being published. It was published last October in an open access Wiley journal, Ecology and Evolution. The link to it is there, but if you just Googled 16 years of breed management is actually enough to bring it up. It goes chapter and verse into the background, the scientific background to the scheme, how it came to be, and the results of the scheme. And the good thing is that it has been independently peer-reviewed. It's not just Andy Dell or the authors saying it. It's actually been independently peer-reviewed and verified that you know, we have actually managed to achieve what we say we achieved through pedigree-based breed management strategy in the breed. So there you go, folks. Thanks for bearing with me. I can see there are at least 11 questions have built up within the chat, but can I just take the opportunity of saying thanks for sticking with us? I know it's been quite a long talk, but also thanks to the Society for, first of all, giving access to the PopRep analysis. The Rare Breed Survival Trust shared it back with me to help put some meat on the bones for this talk, and thanks for the opportunity to talk to you. And probably best now if I actually stop sharing my screen so I can see some faces, and happy to take some of the questions that are in chat or throw it open to the floor. Lots of faces. Jill, I think chat's been pretty busy.
It has. A question here from Rodney Reed, who says, if you have 2 stud books, why are you thinking it's not a good thing to add them together into one for the Sparks system?
Right, historically that happened with the Yorkshire Coach Horse Society. The actual number of horses within that stud book back in the 1920s was relatively small, but it did show the impact of bringing them together. Breeders would say that were those horses Cleveland Bays? Well, what's been accepted is that some of them were 1st generation crosses Cleveland Bays, and it was effectively equivalent to a grading up scheme in amalgamating the stud books. But essentially the breed has been closed since the 1930s for way over 100 years. We maintain our stud books, certainly the Rare Breeds maintain our stud books as essentially closed stud books. Rare Breed Survival Trust doesn't actively support grading up schemes, although it recognises that some breeds do have grading up schemes. I think it's important that you actually understand your breed before initiating a grading up scheme. Do you actually need to grade up? Originally, the grading up scheme within Cleveland Bays was brought about because it was recognised that there were some mares of Cleveland Bay type out there that missed out in that first round of registrations in the first 2 or 3 editions of the stud book back in the late 1800s. There were some that got missed that clearly were of Cleveland type, and they wanted to include them within the ring fence, so they brought them in via a grading up scheme. At the end of the Second World War, when the Cleveland Bay population was down to just 4 stallions existing in the world, they again were grateful of the grading up scheme, but it was female based, not male based. But yeah, it's not had a huge, if you look at the influence of the grading up scheme within Cleveland Bays, it's been relatively small, and very little gets up through the grading scheme into the full purebred stud book.
Okay, the next question is, how would a breed association, NASHA, go about getting involved in the Sparks program so we could build this sort of breeding information to members?
Right. So the first thing to do is to take your stud book and make sure it's digitized, which most stud books are nowadays, and then you can take that stud book data, and if you want to do, you can just put it through PopRep, is as good a place as any to have a look at it and do a state of the nation, because it's only when you understand where your stud book's at to understand whether or not a breed strategy like Sparks is relevant or necessary. But if, having done that PopRep analysis, you decide that you want to look towards something like a Sparks type analysis, a breed management scheme, well, Sparks is sat out there as a really good model. Cloudlines, as I say, is another breed management suite of software that has a built-in Sparks type module within it, although it's not specific to equines. But the more people that came on board something like that, the more closely tailored it is. I'm actually doing some work with a number of breeds at Nottingham Trent University that could potentially lead to a standalone module that could potentially be tapped into by other breeds. But from the Rare Breed Survival Trust point of view, what I've agreed is that certainly breeds that are on the priority list, we will try to make available Sparks if it turns out to be relevant for those breeds. And that's been done at this moment in time on a voluntary basis. But certainly PopRep analysis comes first before you decide whether a Sparks is, or a Sparks type breed management scheme is relevant. Okay, and that's all we have with questions wise on the chat. Okay. Andy, if we throw it open to the floor, if anybody else. Oh, hang on, we've got another one coming. Are the DPS Council looking into utilising Sparks or similar, or is this up to breeders due to restrictions? If so, is anyone currently leading on this? Have you much appetite from breeders? That's not one for me to answer. I think that's for yourselves to answer.
Yeah. Janet, do you want to take that one?
Well, I was going to actually pass it over to Joe, but it is something that we are actually looking into at the moment, and council are, you know, sort of discussing it at meetings, because it is something that, you know, we have got consideration for. But we're only in the infancy at the moment. So watch this space would be what I would say, unless Joe has got any more to add. With
just unmute myself. No, I think we are looking quite seriously at it, really. But it's a slow process. We have to put the information out there for breeders and let people get used to the idea. And then hopefully Andy might have a look at how long it would take to produce sheets for them, and you know, how long it would take to get on board. But I think it would be a very good tool, and I think people should be given the chance to use it if they want to.
Yeah, I think the first thing is to do that PopRep analysis again. Let's just see where we're going. But it's exactly the same data that you use for the PopRep analysis that we use to actually produce the Spark sheet. So, you know, one can go hand in hand. and yeah, it's not impossible to put together, but it tends to be a little bit seasonal. And if suddenly a breed society put its head over the fence on the 1st of March and said, can I have it tomorrow? Then the answer is going to be no. It's better if we actually do it in a planned way. I mean, the pop rep analysis doesn't take long at all. That could be done almost tomorrow. But in terms of putting sparks together, I mean, I've been doing it with Cleveland Baits for donkey's years, but at the start, you know, there are always hiccups. It's not directly tied to Jennyped, to grassroots, and it doesn't suck the data in directly from grassroots. There are always hiccups, and you know, there's the inherent problem of, you know, if people lose horses in a year, they don't always turn around and tell the breed society, and the analysis is only as accurate as the data, and eventually find out that horses have died and they shouldn't have been included in the analysis and things like that. I think with the Eriscape ponies, there's a real problem at the moment with a filly foal that was mistakenly registered in grassroots as a male. The analysis has been done, males versus females, and it's screwed everything up. So we're now in version 4 for this year, and that's, you know, 4 years down the line of sparks. But yeah, it's not always as straightforward as it could be.
Yeah.
And thank you, Andy, and we will be in touch.
That pop rep work was done, actually, the last few years. We did do that with the red breeds.
Yeah, I think
thought they'd shared it with you, which
I got the 2000, and the pop rep that I got was from 2017 data, and I think it would be worth just running the most current one just to compare the two and see.
That's
where we're
right,
going.
Which we did do. At least I asked them to, and I rather thought we had.
You may well have done, but it may. I think it might have just been the only one that Tom Blunt actually had sat on his PC to share with me.
Fair enough.
Can I just say thank you so much as a human geneticist and also a canine geneticist and a medical scientist. This has been fascinating, and thank you so much.
You're more than welcome. I think that was Val speaking there, was it?
Yeah.
Yeah. Thanks, Val.
Yes, thank you, Val. Has anybody else got any questions for Andy?
What I will say is it's one of those things where people go away, cogitate, and perhaps come up with questions later. So if they want to fire them back via, is Jill the best?
Jill's sort of your point of contact. So
yeah, if they fire them back via Jill, via
Jill's
email or whatever, then Jill's got my contact and I can have a look at them. Yeah, so I'm more than happy to address questions later, or Jill can fill them out to wherever they're relevant, really.
So well, on behalf of everybody, Andy, I would just like to thank you for giving up your time to sort of enlighten us this evening, and also to thank everybody that's actually, you know, taken part tonight, and hopefully, you know, they've left with more knowledge than they came with, and which I'm sure they have. So, you know, thank you everybody, and you know, hopefully we've all learned a little bit from tonight. So, you know, thank you, and there will be more.
Can I just say thanks again, folks, for sticking around. There's still an awful lot of people in this chat, and we've been on the go for well over an hour and a half now. If you want to blame anyone for that, I have to point. This was the mayor that started it all. That's Caroline and Imber. She's nowhere. That was our foundation mayor. She's no longer with us. It was her fault, not mine.
Well, I think we need to thank her, Andy, not you know, blame her.
Yeah, she's. That was her legacy to the breed. She left. I think she left 7 pure foals on the ground. But no, yeah, I think that's true. But yeah.